There we go. Right. Hello, everyone. Welcome to session 12 of BGA24. This session is going to be on the manual curation of bird microchromosomes with HiC data with Tom Mathers. And now there is an echo.
I've unmuted, it might be me. Sorry.
No, I think it was me because I've got the YouTube stream going on in the background.
Okay, cool.
Right. Okay, let's start it again, shall we? Hello, everyone. Welcome to session 12 of BGA24. This session is on manual curation of bird microchromosomes with HiC data, and the session leader is Tom Mathers. Take it away, Tom.
Right. Thanks, Damon. Hi, everyone. Welcome to the workshop. I'm just going to start sharing my screen. Hopefully that should work. It's asked me to send a request. Are you seeing Are you seeing the slides, Damon?
I am indeed.
Right, okay, yeah, sorry about that, everyone. All right, yeah, thank you. Welcome to the workshop. So we're going to talk today specifically about how to curate bird genomes, and in particular, the curation of bird microchromosomes, which in PacBio hi-fi assemblies are particularly challenging. So the idea of this workshop is really to focus on the biology of the microchromosomes, how we curate them in PreText. It's not so much about the nuts and bolts of how we use PreText. But if you look in the Discord channel, I've posted an information sheet with a bunch of links to various software tools that are used to create the maps for the workshop, and instructions for how to process an assembly using PreText. So hopefully they'll be useful to help you with those aspects. So I'm Tom Mathers. I'm a genome curator at the Sanger Institute. Also involved in the workshop is Joe Collins and Dom Absalon, who are also very experienced genome curators. They're going to be on hand to answer questions on the Discord. If anybody needs it, I think we'll be able to have breakout rooms available if somebody needs some one-on-one support to help them during the practical session when we're going to be having a look at curating a bird using PreText. So yeah, as I said, please post the questions on the Discord, and Joe and Dom will monitor those. And check out the information sheet that I've posted on the Discord as well, which has lots of useful links and resources. And the first thing we need to do is launch our Gitpod pages, because sometimes that can take a little while to start. This is what we use for the practical session. So if everybody could go to to launch the let me just show you the page. So I think hopefully you'll have this link on the BGA page and open Gitpod here. And this will allow you to start the environment. Click continue, and you should be this will start launching the Gitpod environment, which we'll use later on. It's important, and Damon's going to remind us about this, that when we're using the Gitpod, we have to every 29 minutes we have to interact with the terminal, or otherwise it times out the Gitpod instance. So Damon's going to remind us to type something in our terminal windows when we need to do that. But I'll hopefully remember that as well. So that Gitpod should now be started, and I'll just go back to my presentation. Okay, so hopefully you've all been able to launch the Gitpod. And I'm just going to give a little overview of what we're going to do in the workshop. So I'm going to give a brief overview of the basics of the curation pipeline, an assembly pipeline that we use at Sanger to generate bird genomes and other genomes of various different species. I'm then going to talk a little bit about the biology of bird microchromosomes, because it's very important to understand the biology of the chromosomes when we're trying to interpret the HiC contact maps. I'm going to talk a bit about the challenges of microchromosome assembly, why they're so tricky, and then introduce some strategies and techniques that we've been developing recently to help curate bird genome assemblies as quickly and accurately as possible. Then we'll launch into the practical session where we have a go at curating a bird map using the approach that I've talked about. And if we get time at the end, I want to cover a little bit about nanopore assembly of bird genomes and how that might help, and how that can tell us a bit about what's going on with why these bird microchromosomes are so difficult to assemble. And we have another nanopore map as well, which we can look at later. if we get time. So yeah, questions on Discord, and then please jump in if you want to ask anything as well. You're welcome to unmute and interrupt me. Okay, so genome assembly at the Sanger. So this is the pipeline showing the assembly process that pretty much all the genomes go through that come through Sanger. Predominantly, we're assembling genomes with PacBio HiFi and HiC reads. Often we're generating phased assemblies using the PacBio and HiC data with HiFi SM in HiC phasing mode to create two haplotypes. And those assemblies get decontaminated and then passed through to us in the curation team for curation. And we do that using PreText. They then get released on the ENA at the end. This slide shows an overview of the PreText curation process and the associated scripts that we have for running, for generating new assemblies after you've loaded your map into PreText. All these tools and scripts are listed in the information sheet that I've given in the Discord, so you can get an overview of how to run those there. But basically, we take a draft assembly file. We generate a HiC contact map after aligning the HiC reads and generate this PreText map, which is where we do curation in a program called PreText, which we'll have a look at later. We then make all the corrections in the assembly using this tool, paint up our chromosomes, and then generate a new AGP file, which describes the changes in the assembly. And then we use a script called PreText to TPF, which processes this AGP file, and then another script called multi-join, or there's a different one called rapid join we use, which then takes this curated assembly file and generates a new faster file of the assembly with our chromosomes in it and the contigs that haven't been placed as well at the end of the assembly. So that's a very brief overview of how it works. I say the workshop isn't specifically aimed at doing this, more about the details of curating birds, but check out the links if you need more information on that. Okay, so bird microchromosomes, a bit about those. Okay, so bird genomes are organized into macro and microchromosomes. This picture here shows an image of of chicken chromosomes in the cell. And you can see there's several large chromosomes and then small dots which correspond to these microchromosomes. And these microchromosomes can be further broken down into sort of medium-sized microchromosomes, which range from about 20 megs to 5 megabases, and dot-like chromosomes, which are all less than 5 megabases in length. And this compares to the much larger macrochromosomes. And it's these dot chromosomes, these tiny ones, which are particularly difficult to assemble. Microchromosomes have some interesting biological features. So this cytology plot here shows it shows chromosomes in the cell stained with DAPI, and then there's another slide figure on the right-hand side showing them stained with a fish probe, which is associated with CPG islands, which are associated with gene expression. And so what we find originally when microchromosomes were discovered, it was thought they were just kind of repetitive and not very important, but actually they're extremely gene dense. And this staining with this CPG island probe shows the density of genes on these small dot-like chromosomes. They have a lot of genes on there, so they're quite important. And often these chromosomes represent a relatively small amount of the total assembly size, but have a large number of genes. So in the cuckoo, for example, the 10 smallest chromosomes represent about 1% of the genome assembly, but they contain 10% of the protein coding genes. They have interesting features in the nucleus as well. So with HiC data, we can model the position of chromosomes within the nucleus. And we find that microchromosomes tend to associate with each other. And 3D modeling of the HiC data suggests that they actually cluster within the center of the nucleus. And there's some experimental data which shows this as well. So here we have an image of a falcon nucleus, and it's been stained for a chromosome, which is a fusion chromosome with a large macrochromosome and a small microchromosome. And the staining shows that this fused chromosome still is found in the center of the nucleus, even though it's now a large chromosome. So it's likely that there's some features of the microchromosomes themselves actually promote this clustering in the center of the nucleus rather than it just being a size-dependent thing. And this clustering of the microchromosomes together and in the center of the nucleus has a very distinct signal when you look at the HiC contact map, and we'll have a look at that in a bit. They're also evolutionarily quite conserved. So across birds and reptiles, we find conserved microchromosomes, and they're highly conserved within birds. So this is useful in terms of we can use whole genome alignments or conserve genes to identify microchromosome scaffolds when we're looking at the assemblies. But what we find is when we get these assemblies through our pipeline, we find that these microchromosomes, and these particularly the very small dot-like chromosomes, they represent this tiny amount of the assembly, but they represent the vast majority of the curation effort. And they are often highly fragmented in the assembly, and the microchromosome contigs are lost within the repeat shrapnel contigs at the end of the assembly, and we have to find them. So this is a pretty typical assembly of a bird shown as a HiC contact map in PreText. This one is the pink-footed goose, and we use this as an example throughout the talk. So here you can see the large macrochromosomes on the diagonal. This is the genome assembly here. And then the heat map in PreText shows the contacts, HiC contacts, either within a chromosome or between chromosomes. And you see the large chromosomes down here. Now, if we kind of do a typical assembly curation, we can paint all these up and have a look at it, and we can really easily identify about 28 chromosomes in this assembly, just from a quick look through like we would normally do in a curation for another species, another type of species. But we have a karyotype for this pink-footed goose, and it turns out that we should expect to find 40 chromosomes, or well, 39 autosomes, a Z chromosome, and a W chromosome in total. So what that means is that we've painted up these obvious chromosomes here, but we're left with this shrapnel content. So all this high contact, these are all individual scaffolds or contigs in the draft assembly. The dense, darkly colored HiC contacts show that they're interacting with each other. They probably have quite similar sequence. So this is a lot of repeat content at the end of the assembly. We call these the shrapnel contigs. But somewhere in all this assembly debris, there's hundreds, if not thousands of contigs here, are 13 additional chromosomes, which we need to find to complete the karyotype of this bird. And so that's obviously quite challenging to find them. So it is possible, but we use various techniques to try and fish them out. So this is the same assembly, but after it's been curated. So here we see at the end of the assembly, there's these very small chromosomes, which are down to about 150 kb in length, some of them. We can see this orange colored HiC background signal in this map amongst these small chromosomes. This is this elevated signal of contacts between the microchromosomes, which is caused by their close position to each other in the nucleus. And we use this off-diagonal gold signal to try and identify contigs in the assembly to place either as individual microchromosomes or join on to other small microchromosomes. And you can see here, so these other ones which are below this last chromosome here, you see this darker signal here on these contigs. These are hitting multiple contigs across the assembly, and these are probably repeat sequence. And you can differentiate the repeat sequence from these microchromosome contigs by the lack of this HiC background signal. So, for example, hopefully you can see my mouse, but if I'm pointing to a contig down at the bottom of this piece of the map, and if you go up the diagonal here, you can see there's very little background signal. It's much lighter, whereas if you compare a microchromosome to another microchromosome, there's this orange signal throughout compared to the other micros. And so this is a repeat contig, and these ones with the higher background signal are likely our euchromatic parts of microchromosomes. And it's important to stress that in a lot of these assemblies, we do find these contigs which kind of stand out on their own, or scaffolds that stand out on their own and look like individual chromosomes, but it's quite likely that they are representing fragments of an assembly rather than the full length of the chromosome. But often this is the best we can do with the data we've got. So likely some of these repeats belong to some of these chromosomes, but they don't map with sufficient uniqueness to be able to join them together. Overall, this process is really time consuming, and anybody in the workshop who's actually had a go at curating birds will know that it's difficult to piece these back together. And often we don't get the correct karyotype. We can't find all of the chromosomes. So at Sanger, what we've been trying to do is trying to generate some tools to help us get to this sort of level of curation more quickly and more accurately. And so what I've been doing recently is developing a couple of things, which I'm going to share for the workshop. So the first thing we've done is developed a tool called Microfinder. So the idea of this program is that it takes a draft assembly of a bird and it uses miniprop to align a set of conserved microchromosome genes to the assembly. And then for every scaffold in the assembly, it will count the number of microchromosome genes, and it sorts the assembly by the gene count, by the microchromosome gene count. And what this does is this allows you to fish out content from the shrapnel contigs at the end of the assembly, which then become candidate microchromosomes, which you can curate. And this gene set was developed from a set of 12 expert curated genome assemblies at Sanger, and we include in the gene set are things which are just located on microchromosomes. So this means that when you get I'll show you in the map later, but when you get random bits of larger chromosomes which need placing onto macrochromosomes, they don't get brought to the front of the assembly because they don't have microchromosome genes. And it allows us to just fish this content out of the shrapnel more quickly. And then what we do is we order the assembly by these gene counts and remake the PreText map using this ordered assembly. And that allows us to curate them more quickly. And that's now applied to all of the birds that come through the Sanger pipeline. Second thing we do, now this is fairly new, so I've been working on this in the last few weeks, but we've had some nice results, so I've decided to include it in the workshop. So, I've skipped ahead of myself. This is just this is a slide showing one of these ordered maps from Microfinder. So here you've got the macrochromosomes, and then right at the beginning of the assembly, you've got lots of very small contigs now. And this is a zoomed in view of those small contigs. And these correspond to candidate microchromosomes. So this is we use this as a starting point for the assembly curation of the micros. So for example, here, this might be one that's on its own. We'd look across. There's another piece here, which we can see off the diagonal, which we might join up to it. And we go through these, and this builds up a scaffold of each of the microchromosomes, which we can then maybe add in repeat content later if we can see some uniquely mapping repeats. But I'll show you that in the practical later in more detail. But we found it quite useful for speeding up the curation process. And in this picture as well, you can see this elevated gold background signal. So you see, for example, if you compare this larger microchromosome here to one of these other candidate scaffolds, you'll see this elevated background signal. If you compare here to this is to the right is this large chromosome. You see this is much whiter, this background. So there's less contacts between this scaffold and this scaffold compared to this scaffold and this scaffold. And this is a classic signal we see of micros where they're associating with each other. Okay, so yeah, so the other problem we have is that because the microchromosomes are so tiny, an issue with PreText is that the resolution of the PreText map scales with the size of the assembly. So the bigger the assembly, the less detail you get when you zoom in. And with microchromosomes, we find that when you zoom in on the assembly, this is kind of like as much detail as you can see often, where you have a very tiny scaffold. It looks like a blob. You can see that it's something that belongs on its own and it shouldn't go with another chromosome. And you can see it doesn't belong, it's not a repeat. But you can't zoom in far enough on this individual chromosome to really see, are there any errors within this small chromosome? And so that limits the accuracy of the curation that we can do. So to solve this, what I've developed, this is brand new. So what I will do after the workshop, we're in the process of generating a bunch of scripts to make this easier for people to do. And I'll email round information on that once it's sorted out. But at the minute, we're kind of doing it roughly by hand. But the idea is you take your draft assembly, that could be a single haplotype assembly or a phased diploid assembly. We run Microfinder on each of the haplotypes to order the assembly. And then we take that ordered assembly and do an initial curation. We're calling this a first pass curation in PreText. We then remake the curated assembly FASTA file, split out all the scaffolds which are shorter than 20 megabases into a separate FASTA file, remake that map, and there's a tool called Curation PreText, which is a Nextflow pipeline which enables you to generate the map and all the information tracks at the same time very easily. So we use that to regenerate the map. And then we do a second pass curation on just this reduced less than 20 megabase scaffolds assembly. And this allows us to zoom in great detail on the individual microchromosomes and correct errors. We then remake a new FASTA file for the micros plus all the shrapnel contigs that we can't place. We rename those chromosomes so they follow on from the original chromosome naming in the first pass curation, and generate a new assembly, new curated assembly, which we can then run various QC checks on. That's the overall pipeline that we use, which we're always starting to use now. This is quite new, but it does seem to work quite well. So I'm going to show a couple of examples. So this is a single microchromosome in a full map. It's on the larger end of the size of microchromosomes. It's probably 4 or 5 megabases in length, this chromosome. And this is a zoomed in view of the full map, the full size map with all of the chromosomes included. This looks pretty smooth, quite nice. HiC is fairly consistent along the chromosome, which is a good sign. It looks quite nicely curated. But this second picture shows the map remade when we've just included scaffolds less than 20 megabases in length. In practice, this gives it, we actually make a map for about 300-400 megs of content using this method typically. And this is the same chromosome in the remade second pass map. And you can see it's a bit fainter, but you can see there's a lot more detail on the diagonal. And we can actually zoom in on different bits of this chromosome to look for errors. And if we zoom in on the middle of the chromosome, so this region I've highlighted here is the center of the chromosome, which is the same as this region on this part of the chromosome in the original map. But when we zoom in this second pass map, we can actually see some very obvious misassembly which needs correcting, which is completely invisible in the original map. So here you can see there's a small piece of sequence which needs to go. There's a break in the HiC context here. This chunk here needs to go across and up to this location where the HiC signal is indicating it should be placed. There's also a couple of haplotypes in this assembly where pieces of this haplotype need to be moved into the second haplotype or removed into the haplotypes bin and removed from the assembly. So that's one example where you can see some clear errors which are basically invisible in the original map, but by making this reduced map, we can get much more detail of the individual chromosomes. And this can actually reveal some quite shocking things. So this is a different bird that we've been curating. This is actually a work in progress curation, and this is the one we're going to use for the workshop when we do the practical session shortly. So this has been curated with the first pass curation, and this chromosome here, this is a 2 haplotype map. So this is hap1 and this is hap2 of what looks like a single chromosome. So we can see this very dense HiC signal joining all these contigs together. This is fairly typical in that you often can't see a huge amount of detail in these maps for the very tiny chromosomes. So this looks like, to all intents and purposes, a single chromosome in the bird assembly. And this is the corresponding chromosome in haplotype 2. But when we zoom in, when we remake this second pass map with the scaffolds shorter than 20 megabases, the extra resolution that gives us allows us to see a huge amount of extra detail on this chromosome. And actually what it looks like, rather than just being one single chromosome, this seems to be multiple small microchromosomes which have been joined together incorrectly. And when we made this original chromosome, this was made from very tiny contigs which were fished out of the shrapnel by Microfinder based on conserved microchromosome gene content. And probably because of the amount of interactions between the microchromosomes, it makes it look like a single chromosome when you're zoomed out. But when you get this extra detail, you can see that there's lots of assembly error, and actually it can be rearranged into 5 chromosomes rather than 1. So here we can, so I've done a quick rearrangement of this, and you can see there's 1, 2, 3, 4, 5 chromosomal units from this one scaffold. So by doing this 2-pass curation, we get this huge amount of extra information which we can use to make a more accurate assembly of the micros. And so this is I've only been playing out this for the last couple of weeks, so it's kind of brand new, but we're going to have a go with this during the workshop now. Okay, so we hopefully have got our Git pod launched. Please post in the chat or speak up if it's not working for you. I want you to, I'm going to just stop sharing the presentation. And if we go into the Git pod, So if you've all launched, I don't know, Dom and Joe, has everybody been able to launch? Has there been any problems with people getting it to work, or are we okay to carry on?
Nobody's commented.
Okay, cool. Right, so to launch them, oh, actually, we need this slide. So we want to launch the Git pod and go, and in the terminal window, you type PreText view, and then add an ampersand at the end, and that will launch an instance of PreText, which is what we're going to do the curation in your window here. And then in PreText, if you click on Load Map and go to, where are we? Workspace. So click on Computer and then Workspace, and then Maps and Save States. And we want to click on the BFA10 assembly here, and then load the file, which is this file here, BFA10 2_HR PreText. If you load that, click on that, it should. There we go. Okay, so this should load your map into PreText. So this is the map we're going to use as an example to have a go at curating it. And what I was thinking would be good is I'll basically talk you through how we go through a curation using this map, and then you can curate it alongside me, and then we'll have a bit of a break where you can have a look yourselves, play about with the map, and ask any questions about what we've been doing. What I'm going to do is switch to my own, because it's a bit laggy. This is the same map, but just loaded up locally for me. So, Dom, Joker, is this showing up on the screen now for everybody in the Zoom?
Yeah.
Okay, right. So, what we've got here is this is the microfinder ordered map for this bird, and the first thing you notice is it's a phased assembly, and we've got both haplotypes included in the map for curation. So the first part of the map contains all the hap1 scaffolds, and then the second half of the map contains all the second haplotype scaffolds. So these individual ones here correspond to large macrochromosomes. This is hap1, and then in the second haplotype, this is hap2. So if you look along the diagonal, and in PreText, hopefully, you can go into edit mode, and you can use the middle button of your mouse to pick up a scaffold and move it around the map. And this allows us to make edits in the assembly. So when I curate a bird, the first thing I like to do is, especially in a merged map like this where we have both haplotypes, is move all the haplotypes across. Spacebar flips things, right click to drop it, and we move all the scaffolds into across. So we are basically joining up hap1 and hap2 so we can see them next to each other. And because we use multi-mapping reads in the creation of these PreText maps, we can see very clearly where hap1 and hap2 agree with each other. So off the diagonal, you get this diagonal line, and this shows where the sequence is very similar between haplotype 1 and haplotype 2 on the contact map. And so what I'll do is move around the map, and I'll place all of these haplotypes together. So we're just moving hap1 to hap2 for each chromosome. And what we find often with bird genomes is that especially for the micros, often you don't get a complete representation of a single chromosome in one haplotype. It might end up, because it kind of drops out, you might end up with a piece of like part of the chromosome represented in one haplotype and the other part of the chromosome represented in the other haplotype. So what we tend to try and do is make one good chromosome out of the two, at least one good chromosome out of the two haplotypes. Obviously, if we get good representation in both haplotypes, we'll curate both, but it just depends what we get. So I'm just going to move these along here and line them all up. Like this. So this is basically just matching up the two haplotypes of the assembly. And for this bird species, so we know we often look for karyotype information for the birds that we curate, so we have a rough idea of how many chromosomes we're expecting. So for this bird species, we know we don't have a karyotype for the exact species, but we know that close relatives have 39 to 40 chromosomes. So that's the number we're aiming for, roughly, but we'll see what we can see in the HiC data. Okay, so I'm just going to finish moving these across. This individual is a female, so we should expect to see the sex chromosomes in this map as well. So here, there's a large chromosome here, which is only partially represented in haplotype 2. So this is likely the Z chromosome, and there's other content here which corresponds to the W chromosome, which is smaller and more repetitive. And this region that's shared is the pseudoautosomal region of the Z and W chromosome. Okay. So just moving, we're joining stuff together based on the HiC contacts indicating links. So for example, here we zoom in, we can see an off-diagonal contact here linking this scaffold to this scaffold. So this piece should be flipped like this. And move this, and then if we look up the diagonal, we can see more contacts here. So we grab this, move it to the end, lightly links like that, and we can add in content. I'm just going to finish moving the two haplotypes across. And then we'll start doing the actual curation of individual chromosomes. Okay, so what I find important to do with birds is often what we're doing when we're looking at the micros, we're trying to find additional content to place in them. But to do that, we're looking for scaffolds which are kind of on their own in the assembly. So here in the shrapnel, you can see this is what we call the shrapnel, which are all the short scaffolds which are left over at the end of the assembly. These are often containing a lot of the repeats in the genome. So for example, here you can see a piece of sequence where it's got a lot of HiC contacts with various other chromosomes, various other scaffolds across the map. So here it's hitting here it's hitting this one. And this is indicative of this just being some repetitive sequence. But for the chromosomes, you'll see on the diagonal, we'll see what we're looking for is more euchromatic type signal. So this is where we see something that's more on its own. It's only hitting to like one place in the genome. There's on diagonal contacts here for this piece. There we go. And then if we go back to the front of the map, so now I've placed most of the individual scaffolds with their haplotype pairs. There's some obvious areas we'll go back to. And then here, at the beginning of the map, is where we have our microfinder ordered content. So these are the additional candidate microchromosome scaffolds. And if we look into the other haplotype, there's a bunch of stuff over here as well. This is the start of hap2. Pick these up, move them across, drop them down. So this is all scaffolds which are potentially microchromosome associated. So if we go down the map, what I like to do first though is place as much content as you can from the shrapnel into the main chromosomes, because that means, because otherwise you can get stuff that looks like it might be an individual chromosome, but actually it just needs placing into one of the larger chromosomes. So for example, here, we look down the map, I'm looking for off-diagonal contacts here. You see that this piece of sequence here has got a lot of contacts with this chromosome. And if I zoom in, you have one. There we go. So, and if we look here, this looks like it needs to flip. So if you look down the diagonal here, there's a break in HiC contacts. Then the end of this contig here has got high contact with the start of this contig. So we want to lift this up and flip it. And then we can look in hap2, we've got the corresponding region in hap2 of this chromosome. So we're gradually building up chromosomes in the assembly using this method. And what I can do now is I can say, is there any more content that needs to go into this chromosome that's located in among these shrapnel contigs? So if I move across the diagonal, I'm looking for extra sequence. And here I can see a scaffold here. This is scaffold 289. It has a lot of contact with this chromosome, but it's quite unique to that chromosome. And if you look further up the map, it's not going anywhere else. So pick it up and place it. This is, well, we generally try and keep content within. So with these merged assemblies, we have the HAP1 scaffolds are all labeled HAP1 and the HAP2 scaffolds are all labeled HAP2. That's where the sequence has been binned by the de novo assembler. Sometimes you get phasing errors and you need to move content between the haplotypes, which we do. But as a starting point, we keep it within its binned haplotype and then change it later if we need to. We can leave that there. We have extra information in the map. So this is at the minute just looking at the HiC map, but we can press U, and then this allows us to load up additional tracks that have been generated. So we have a coverage track. So this is the PacBio hi-fi reads mapped back to the assembly. And we have a gap track. We can change the colors just by right-clicking over here. We can change the colors. So these are all the gaps in the assembly. And then we have a telomere track, which changes the color to purple, which indicates where the telomeres are in the assembly, or where candidate telomere sequence is in the assembly. So I'll just turn the coverage off and the gaps off for now. We'll leave the telomere on. Okay. So what I would do is I would go through this assembly, place as much as I can within each individual chromosome that I can see. This looks like it's a piece of the sex chromosome. So keep moving stuff around. Now we'll go back to our microchromosomes. So these are basically the smallest chromosomes which we want to try and identify. So one thing to know is, so when Microfinder has ordered the assembly like this, what we often see is it finds very tiny contigs in the assembly, which, because of the resolution issue with PreText, it doesn't always render completely properly. So if we press G on the keyboard, this toggles grid lines in PreText. And these grid lines, which are by default in gray, are separating individual scaffolds in the assembly. And then within a scaffold, you might have contigs joined together. You see there's lots of gaps here. But the gray lines are showing gaps between scaffolds in the assembly. And with the tooltip on, so at the minute, when I hover over a scaffold, it's telling me the information near the cursor is telling me what the scaffold ID is of that particular piece of the assembly and the position along the scaffold. If you can see the numbers moving, this is the position along the scaffold. Now in this assembly, because there's so many tiny scaffolds, PreText isn't correctly rendering all of them. So here we can see this is scaffold 40, and it looks like scaffold 40 ends here, where this gray line is. But if we hover over, we can see that actually it's 10 or so very tight. So you see the number change, so it's going from 33, 31, 29, 27, 26. So these are actually, and we can, if you edit mode by pressing E to pick stuff up, middle button, these are actually individual scaffolds. I've not broken this scaffold. I've just picked up additional scaffolds here. So this is just something to watch out for. Because they're so tiny, it doesn't render them properly. So there I've picked up the scaffold 40 and left behind this bit of content. Okay. And so what we're looking for is basically joins that we can make to join some of this content together into individual chromosomes. And if you look across the map, you can see that these candidate microchromosome scaffolds are fairly unique in the sense that they don't look like they go anywhere else. They belong with each other. And if I just go into the shrapnel contigs, and they are hitting some of these repeat-like contigs as well. So we might be able to drag in extra sequence into these as we start to build up the chromosomes. What I'm going to do is move them down to the end of the map now. So I'm going to pick up, so to pick up multiple contigs, you hover over at the start where you want to start picking up, hold down the middle button and drag down. And I'm going to pick up all of these small contigs and just move them down to the bottom of the assembly here. Okay. And what you can see in the if you look at the background sequence, I'm going to move, this is the Z chromosome, we have the W chromosome here, move that out of the way. Up there a bit. Now what you can see is this elevated background signal that I mentioned earlier. So this is what we look for when we're looking for microchromosome associated scaffolds. So if you look at the larger chromosomes, the background is very white, indicating a low level of background contacts between these chromosomes. But when you go down to the smaller chromosomes, you can see that the background is much yellower. There's much more interchromosomal contacts between these scaffolds. And this is because of, as I said at the beginning, the biology of the microchromosomes. They group together in the nucleus, and this background signal, so that gives us this elevated background signal because they're just touching each other more in the cell. And we use this background signal to try and identify microchromosomes. And basically, if you had a small contig on its own here, but it didn't have this dark yellow off-diagonal signal, you might be suspicious of that one and think, actually, should that be something that belongs in one of the larger chromosomes? It's just a bit that's not been placed. So we're always looking for that enriched background signal. Okay, so here we've got, so we can start to build up the chromosomes. So here we've got a piece which is potentially a piece of microchromosome. It's got its partner here, so this is a hap1 scaffold. This is its partner in hap2. Flip it, place it there to start to build it up. There's another piece of sequence here in hap2. This looks like it belongs with this one. So pick it up and make the join. Start to build this up. This piece is hap1, and it looks like it goes at the beginning, at the end of this chromosome here. So basically, we're gradually just building up individual chromosomes by placing some of this content. Like this. And we can also look at the gaps. So some things might break as well. There's a piece of sequence here, for example, that might place in here. Move this one up here. So this one goes with its partner over here. And there's here you can see, so this is part of a chromosome here in hap1 and its corresponding partner in hap2 in the second haplotype. But you can see that the order of the chromosome is different between hap1 and hap2. Let's put on the gap track and see if there's any gaps. So here we've got gaps shown in green. So there's probably some sort of issue with this scaffold. And we can try and fix that. We can check for a telomere sequence, which we don't have. So here, it looks like possibly we want to move this to the end here. So now we've got smooth down here, and it's smooth down here. There might be a piece missing from this here. We'll not go there. So see what we can see. So here we're looking for extra unplaced content that may belong with one of these chromosomes. I can't see anything immediately obvious there. Let's turn the gaps off. Something else. Okay. So piece goes down. So what we can do is we can have a count of what we've got so far. You can see here, here's an example here. This is a slightly larger microchromosome here. And you can see that there's this is a scaffold, you can see by the gray lines. And in the middle of the chromosome, the contacts just drop off altogether. And there's this purple line, which is showing this is our telomere sequence in the middle. So this actually looks like two separate chromosomes which have been incorrectly joined together by the scaffolder. So there's one here and another one here. And there's a gap in the middle, which we can use to break it. So we go into edit mode, pick this up, we can flip it to break it. So right click over the bit we want to pick up, flip to break it, and then we can find its corresponding partner in the other haplotype. So here, just like that. And then we've got another piece there. And this piece, here, flip that. And this piece needs breaking. Yeah. Where's our start bit? Here we are. Okay, so I did that in a bit slightly roundabout way. I'm sorry about that, but I'll just undo. Okay, so we have what looks like two chromosomes. Identify the breakpoint. This bit needs breaking off. Pick it up, break it. And then this piece is another piece of that same chromosome here. You can tell by the off-diagonal contacts, but this is the wrong way around because this needs flipping. And then you can see here that now we're looking for this nice smooth on diagonal all the time. This piece is this corresponding chromosome in haplotype 2 for this one. Move it next to it, flip it so they're in the same orientation. Now we've got the second chromosome, which was originally joined together. So these are hap1 scaffold 97, hap1 scaffold 97, two pieces of that original scaffold, which have now been broken into 1, 2 chromosomes. Okay, moving through. So this piece here, we've got a piece in the middle here which belongs over here. We can pick that up and place it with this piece of W. And just check, there's another set of, so these chromosomes look here. So this is some larger macrochromosomes, larger chromosomes, which have this lower background signal. And if we zoom in here, the scaffolder has joined 3 chromosomes together here. You can see this very sharp drop off in HiC signal here and here. And if we look at the gap track, we can see that these should be broken. So we can zoom in, break this one, break this one. And these are probably joined together. They have this elevated background signal. You can see there's more orange here than compared to the larger chromosome. And there must have been some context which have caused these to be joined together by the scaffolds, but it's an error. And then we need to break the haplotype 2 sequence. Yeah, pick it up, put it together, flip it. That one. So we're trying to build up the whole map really to find all the chromosomes, but we can flip this. So now we've got that one, what was joined as 1 is now 3 individual chromosomes with their 2 corresponding hap1 and hap2 sequences. Okay. So the really tricky bit is obviously these very tiny bits at the end. So we have some good, this looks like a decent one. So I try and group ones which you think are starting to look decent together. So here is one, there's a bit of a gap in this one, but this copy looks not too terrible. And this looks like some sort of repeat, but it's got possibly something in the middle of it, which is more euchromatic. So here there's like a piece of repeat, and then something which is this more smooth on diagonal signal, which has a bit of background. And if we look in the second haplotype, it has a corresponding piece which matches. And we know that it has gene content on it. So this is potentially a chromosome. And then there's all these small stuff which look like they go together. And here, here, this is another case where, because of the resolution of PreText, we've got actually multiple scaffolds here. So 62, 63, 64, 66, and then 68 is bigger. So these, but because this looks like it doesn't join together here, there's a drop off in this context. And there's actually no contiguing join, it's just scaffolds. So this content can be moved up somewhere to go with these ones. Move that piece of it away. And we want to try and build up content. So this looks like it, this is what looks like it might all go together because of this dense background context with each other. Okay. And we can add additional stuff in here. This. So these bits, this is some of the hap2 sequence, which looks like it corresponds to this part of hap1 sequence. We're joining these together. So it's not the easiest thing to do for these very small bits, but we can start to build up what looks like it might be a chromosome. Right. So I don't know if are there any questions in the chat, Joe? I can't hear.
Sorry, no, not that we haven't answered as far as I can see.
Okay, yeah. So it's kind of Pardon?
It was just someone joining that wasn't muted.
Okay. Okay. Right. So what we started to build up a bunch of chromosomes here. So we can paint them up to count what we've got so far. So if we go into scaffold mode, start painting. So we call this one. We'll just count one haplotype to start with. So we think we've got something here which these all look like they go together some way. We'll count from here. 1, 2. And what we want to do eventually, so we basically, when we've counted them all up, we'll have what looks like, correct scenarios here. So this is another one which looks like it needs a flip. Okay, so this looks like probably it breaks off here. Beginning.
That'll be your warning
Okay.
to go into your Git pods and have a click around.
All right, so if everybody wants to go into, so we can just in the terminal just type something, and that should keep it going. Okay, so what I've done though now, so if anybody has any questions, please speak up. It's it's not the most straightforward thing to do, but we've identified some small chromosomes already. We've got a bunch of content here which looks like it might be a chromosome, but it's quite difficult to work out exactly what's going on with it. There's other small bits here. What I'm going to suggest now is you go into the Git pod. So in the map, we can also, what I've done is I prepared a save state of this assembly once it's been curated. So if you click on load state and go into computer, workspace, maps and save states, and then click into BFA10 and click on the BFA10 to _HR pretext.savestate1. And this should load up basically a semi-finished curation of this assembly. So here, what I've done is all the chromosomes have been put next to each other with their HAP1 and HAP2 partners. They've been painted up as HAP1 and as individual chromosomes. They've been labeled by haplotype, so that when they get spit out into separate haplotypes. And at the bottom, we can see what we think of the microchromosomes that we can find in this assembly. So you can see individual scaffolds here. So this is our elevated HiC background. Here's a chromosome. Here's the second haplotype, another chromosome, second haplotype. And if we zoom in, so because what we would normally do as we're curating these, it would normally take quite a long time to put all this together. So the curators design would typically take at least a couple of days to do a bird normally. So you kind of gradually build it up, looking for these scaffolds that look like they belong together using the gene content. But this is like a shortcut. So we've got this save state which we can use. Okay. And okay, so you can have a look through that. And so in total, we've identified 66 chromosomes, so 33 haploid chromosomes, 66 in total because we've got our pairs of chromosomes, including the sex chromosomes, Z and W. We've painted up W and Z, which are here. So these are some examples of some of the smaller chromosomes here at the end, which are our micros. Now, this is where I would then say that looks pretty decent for what we can do in this scale of map. So we've got the smallest ones down here. This is very small. Okay. And what we would then do now is take this as our first pass curation and output a new assembly file from PreText of each haplotype, regenerate a FASTA file for each haplotype, and then split out the small stuff from the big stuff. And in the talk, I showed you this chromosome here, which is all this small stuff, which looks like it generally groups together, but we can't see exactly how it goes. You know, we can't see a lot of detail in this at this resolution, but it looks like they all belong together. If we now if you go into on the Git pod, load map, and click on the file which is merged first pass 20 meg HR_pi.pretext. So this is a map just of the smallest chromosomes in that assembly, which are all the scaffolds less than 20 megabases, which have been remade. So now we have, these are our candidate microchromosomes, which have been put together in the original map. But now we've remade the map. We can zoom in much greater detail to correct additional errors. So I'm going to move down the map and have a look at these chromosomes and look for errors. And so here, this is the one that I showed in the slides earlier. So if you zoom in, so normally you wouldn't be able to see an error like this in the lower resolution map. We turn on the gap track, we can see. Right. So here we can see this piece of sequence here. We can zoom in to quite high detail. There's two gaps either side of this piece. It looks like it picks up and moves into here and flips. There, it's now been placed, and it looks like there's a small piece of sequence here, which looks like it's possibly a haplotype. So this is a piece of the second haplotype, which has been assembled into the first haplotype. So if we zoom out, this is still a merged map, so we can look at the second haplotype for this chromosome. We move across. And we pick up the second haplotype, move it adjacent. Okay, drop it in. And if we look across, so we have this, if we zoom right in, see these parallel lines off the diagonal. So this piece of sequence here is very similar to this piece here. And it may look right across. We can check to see if it's already found in the other haplotype assembly. So what I would do is pick it up. Now you can see that it's present here. If you zoom in, there's parallel lines to this part of the assembly, and let's find it in here. It looks like it also is fat, so it looks like it's actually split. So if I drop it, you can see part of it is mapping to here, and the other part is mapping over here. And we might not be able to see it if we turn the coverage track on. Over these regions, the coverage is quite low, so this is probably an additional false duplication, which we can remove from the assembly. Pop it to the end, drop it down, and we can label this up as a haplotype. So in our metadata tags, we can load it up, select it, click it as a haplotype. That means that when we remake the assembly file, that will be ejected from the assembly and not included. If we go back up. And now, so this now looks smoother down here. We've got a bit of possible stuff missing here. There's an extra bit here. So you can see again this small piece, which looks like it may be a haplotype. So we'll look in the other haplotype. Okay, so it's also present in this haplotype as well. The thing to look at here is, if we zoom in, this is about as much as we can zoom in. So this piece here, the coverage over these two regions is dropping by half. So likely this is a false duplication in the assembly again, and it could be removed completely. Because we've got it, that sequence is represented in both haplotypes, but there's this extra piece where the reads are mapped to, and it's reduced the coverage by splitting the reads across the extra content. So this is probably a false duplication that can be removed. And split it out and label it up as a haplotype. Now, if we go through, What I want to do is go down to the smaller chromosomes and see what we can see in the smaller chromosomes. Okay. So this is our particularly problematic chromosome here. So this was labeled up as a single chromosome in the original assembly, but now we've got this extra detail. We can see that actually there's lots of assembly area going on here. And if we load up the gap track, you can see gaps all the way along this sequence. So I'll also bring in the second haplotype. And here we can zoom in. So this is basically hap1 and hap2 of this problematic chromosome. So what we can do now, we've got this extra detail. We can load up the gap track. We start to break this up into individual chromosomes. I'm just going to change the color so I can see it better. So we're looking at, along the diagonal, we're looking for smoothness, and we can see these various drop-offs in sequence, in HiC contacts, and bits which look like they should break or go together. There's also evidence of hap1 being mixed up with hap2. So for example, here, this, oh, that's the wrong bit. There's a gap here, this piece of sequence here. Looks like it belongs to something over here. It's the same. So one of these pieces belongs in a separate haplotype to the other. What I'm going to do is break out the big chunks first. So here you can see this this content here belongs together. It's got a lot of height, a lot of background. I'll pick this piece up, and I'll just move it to the start of the chromosome. There's another piece here which I'll split out. And because this is so jumbled up, we really need to just break it up into bits that look quite reliable, and then start adding content in to make these chromosomes. Another bit here. Move it to the start. And then because we're curating both haplotypes together, we'll want to have the corresponding sequence from the other haplotype as well. So we can start to build these up. Here's one, another chunk which all goes together. And what we can say, we probably might not expect to be able to place all the content that's been identified as potentially microchromosome into defined chromosomes, because some of it will be too small or be too ambiguous. But we can try to build up these units as best as possible. So here's another chunk. So move it to the beginning. And so we're just gradually building up the first haplotype, just with units which might be candidate chromosomes. And we can see along this bit of the scaffold, there's various bits. So this bit, for example, has a high background with this bit. You can pick it up, place it, and it looks like it belongs at the beginning of this chunk here. You see the higher contacts here than at the other end. This bit, it looks like it's a haplotype of this part here. These parallel lines again indicate another part of the other haplotype. So we'll take one of the copies out, move it out of the way. There's another bit here. So this bit here, look at this small square of sequence here. It looks like it belongs with this one. Move it to the beginning. There's lots of very tiny errors in this. So here, another piece that looks like it belongs with that. And we'll check, does that go anywhere else? Well, it's got high contact with the rest of this. Zoom in as much as we can. We'll break the end off this bit. Pick it up, move it over here. Put the other piece. Okay. When I zoom out, I'll take the grid off, take the gaps off. So we have like 1, 2, 3, 4 pieces which are starting to come together. we need to add in a lot more of this content to each of them. And then what we'll do is we'll just put the content from hap2 adjacent to each of those chromosomes to try and get both haplotypes built up. So we can see here there's a big drop off in contacts here. We'll take this whole square, which likely looks like one thing. Move it up. Okay. There's a piece of it here. So this small piece now, where are we? What's going on? So this bit here corresponds to that one. This is in much smaller pieces, this other piece here, so we can pick up bits here. So there's a drop off in contacts there. That's a square of stuff. Move it along. Here it goes there. So yeah, so basically we want to gradually build up all of these small chromosomes. And ordinarily, a lot of this content, because if you hadn't fished it out with the gene mapping, you just wouldn't see it in the assembly at all. It would just be lost in the shrapnel. So these are chromosomes which would have ended up being missing from the final assembly because they're just made up of two small bits. It was too fragmented initially. But because we've identified them with the gene mapping approach, we've been able to bring them into the assembly and then correct them in the second pass. This is still super messy. It needs a lot more work. You could spend days really assembling this nicely. But the idea is we're just build up each of these chromosomes. And if I take the gaps off, so we're going to jump ahead again now. So what we can do is if we go into U, press U, and then what we want to do is load a state, another save state. So I've made another save state of this curated second pass assembly. So if you click on load and then load state, merged first pass, max 20 megabases, click on this one, and then this will show a curated version of this reduced map. So now you can look through. I'm just going to turn off the gaps and the coverage. And I just want to hide the haplotype labels just so you can see what's what. Okay. So what I've done is I've placed each haplotype next to its partner. So here we've got first chromosome, hap1, hap2, second chromosome, and then these have been painted as individual chromosomes. And we go down to the smaller content in the map, you can see, so these all, because they stand on their own in the HiC map, these are all individual chromosomes. And if we go down to the bottom here, we can see that we've been able to curate these very small ones by gradually building on. And you can see the number of grey vertical lines shows just how many rearrangements have had to be made to bring these bits all together. Well, you can gradually build it up. One thing to notice, there's often, this is quite typical, there's a lot of missing content in one of the haplotypes. So for example, here, we've got a chromosome here, which is labeled up as Super 29 in the original assembly. This is hap1. There's a big repeat on the beginning of it. But that repeat isn't present in the other haplotype. It may be there, but most likely it's just not been assembled properly. And then you can see off the diagonal, you see these gaps. So here, there's a bit missing here and a bit missing. So this sequence here in hap1 is just mostly not present in hap2. You can see the second part of it is present there, but this piece here is missing. And if we look at the coverage, it possibly, yeah, it is slightly elevated over this region. So this is probably just collapsed content in the assembly where we've only got a good assembly of one of the haplotypes in this particular assembly for that region. And often you can see this in more extreme cases as well, I think, in some of the other chromosomes. So find something smaller. Yeah, so here, for example. So here we've got, this is one reasonably good haplotype of a chromosome here. You see it's got high background signal with itself, and then you've got this light gold background with the other micros. But if you go all the way up the map, you'll see that it doesn't really, all of this sequence doesn't belong with any other chromosome. It's on its own. So this is haplotype 1. But in haplotype 2, we've only got 3 small contigs of this chromosome, which correspond to a piece of it here, a piece of it here, and a piece of it here. So this is, and if you go along the diagonal in the other direction, you could try and drag in extra content, but I've already done that for this chromosome, and really you just can't find any other content for this chromosome. So we've got a reasonably complete hap1 and a fairly partial hap2. And that often happens with the micros, where in extreme cases, you'll just have a representation in one of the haplotypes, but not both. And that's because of issues with the assembly. So we try and really, we're just trying to do our best with what we've got. We drag in as much stuff as we can using this off-diagonal signal, the gene mapping, but often we can't fully resolve all of the haplotypes. But here, this is given as our 40 chromosomes now. We've broken up the bits that were incorrectly joined together. We've got 2 haplotypes of each chromosome as best we can. And these are very tiny bits. So, you know, you're talking sort of 50 to 100 kb for this one over here. Okay, so now when you've done that, you then remake the assembly file for this scaffolds less than 20 megabases. But at the minute, I'm developing some scripts to rename the chromosome so it can be just placed back in with the large chromosomes from the first pass assembly, but that's not ready yet. So you need to join them back together yourself and rename, and then that gives you your curated assembly. So I don't know if anybody's got, it's a bit kind of, it's obviously a lot to cover in a short amount of time, and it's quite complex, but if there's any questions, I'm happy to answer them. If anything's not very clear, if not, we can move on to the next part of the workshop. Okay, all right, so I'll go out of there.
I do have, it's first a comment saying that it's really impressive to be able to get that level of details, because that's always something that's a bit weird when doing manual curations. Like, I'm not sure, I don't know if it's exactly what I see. So I guess my question is, have you tried doing that? I guess there is downsides to what I'm going to ask, but doing that for other genomes than bird, for which the assembly, you could see that there were issues with the assembly, but it was not clear enough.
To be honest, I just tried this last week on a couple of birds, and I apologize if it's a bit difficult to describe, or it's because it's a bit complicated. But yeah, I think it's a good suggestion. I think we will try this with other things as well, because we often find that we can see things. If we make, say, a HiGlass map of an assembly and zoom right in, you can see a lot more detail. It depends on the quality of the HiC data, really, I think, and the coverage of the HiC data. But I think there is extra information there. The problem we have is when we're trying to curate a lot of genomes quickly, we don't always have time to dig into the HiGlass, try and guess where it is in the PreText map, and make the change. Whereas if we can make these reduced maps for certain parts of the assembly and get higher detail, we can do the normal PreText editing in those, and it allows us to make these changes. It makes it more feasible to make the changes. So I think it's definitely worth trying in other things.
Technically, it will always take longer, because you will see more things, so you will think about more things you can arrange. But that's super interesting. Thanks for
from playing about with it, my initial impression is that, because often with birds, when you get down to the very tiny bits, you kind of saying, well, that looks like it's on its own, and we think it looks like a chromosome, and that looks like the other haplotype of that chromosome, but really you just can't see any detail, or you're kind of guessing a bit too much about what represents one chromosome and if there's any errors on it. With this reduced maps where you can see more detail, I think there's less guesswork, because you can see more of what's actually going on. And so, what happens with the curators often is we'll spend a lot of time where it gets a bit stuck, where you just can't see whether it's one thing or another, and you end up procrastinating over it. and it takes that takes just as long. You can waste a lot of time just really not being sure about where to place it. Whereas if you make this reduced map, there is more to do, but when you zoom, you can actually see where it goes over a different location often, is what I've found so far. And I mean, for example, I think this map, I've remade it now, and there's about 400 edits in the whole map, just of the scaffolds shorter than 20 megs. So there was, in this assembly, there was a lot of extra work to do by zooming in, but I feel a lot more confident about the individual chromosomes and the quality of the assembly of those chromosomes than if I just used the zoomed out map. So I think it helps.
Yeah, I felt very seen by what you said when you said procrastinating, because you don't really know. So yeah, but well, yeah, thanks for the word.
Yeah. Okay, good. I don't know if anybody else has got any comments or questions. Apologies if it was slightly rushed or confusing, but there's a lot of information. What I can do now, I'll jump back to the slides, and I just want to show, I think we've got a bit of time. So where are my slides? Okay. So what I want to do in the final part of the workshop is just talk a bit about, about why these.
Just the alarm for the Git pod. So if you do want to keep using it.
Oh yeah, we might jump back to the Git pod. So if people want to just wiggle it, you can just just type something in the terminal and it should keep it going for a bit longer, because we're going to jump back to it for the last part of the workshop. Okay, so what I wanted to talk about just at the end of the workshop now is looking at why these microchromosomes end up in this horrible state in the first place. Why are we struggling to assemble them with this HiFi data? It works really nicely for the large chromosomes. We get really nice assemblies, but then when we have these tiny chromosomes in these birds, they're really so fragmented, and what's going on? And can we bring in some extra data to try and help us assemble them better, in particular looking at nanopore data for these genomes? So recently there was a really nice assembly published of a near complete chicken genome assembly. To generate this assembly, they used nanopore data and PacBio HiFi, and they used a phased approach, and they basically tried to assemble it with each technology and then use one technology to patch the other assembly, and build up a more complete assembly. And what they ended up with was essentially a near gap-free assembly of this chicken. There's a few gaps in it still. And this is the HiC map of that assembly. You see the microchromosomes are at the end of the assembly here. You see these elevated contacts amongst these small scaffolds. They've put the sex chromosomes at the end there. And if we zoom in on the 10 smallest chromosomes in this nanopore assembly, we can see that we get to see a bit about the structure of these microchromosomes when they're nearly complete. So we see these euchromatic regions, which have got smooth on diagonal signal and this elevated HiC background, and then we get blocks of repeat across the chromosome as well. And a lot of these repeats are found on multiple chromosomes. So you can see here, this bit here corresponds to a similar repeat over here. There's this repeat here. It's found in this chromosome, this chromosome, this chromosome, all the way along. And this shows as well why it's often hard to place the repeats on microchromosomes, because they have a lot of similar repeats across different chromosomes. But also, the thing to notice is the size of these chromosomes. So the smallest chromosome in this assembly is actually 2.5 megabases. Now, if we compare this to a HiFi assembly of chicken, so the most up-to-date HiFi assembly is BGA24, which was done by Sanger and the VGP. This is PacBio HiFi plus HiC data. And if we look at the chromosomes in this assembly, the smallest microchromosome is only 96 kb in length. So it's dramatically smaller. And that's a combination of things. It's not being able to place all the repeat content, because often in birds you find there's a lot of shrapnel left at the end of the assembly, which it hits so many places in the genome you don't know where to place it and you have to leave it out. But also there's something else going on. So here we've got a comparison of the size of these 10 dot chromosomes. In the reddy pinky color here, we've got GALGAL5, the PacBio HiFi assembly, and in turquoise, we've got the new nanopore-based assembly. And in all cases, the nanopore chromosomes are longer, which you might expect, but they're dramatically so in some cases. So, for example, chromosome 38 is like 1/10 of the length in the HiFi assembly as it is in the nanopore assembly. But if we compare the large chromosomes, the macrochromosomes, they're all within like 5% tolerance of each other, so they're much more similar in size. And if we take this high-quality nanopore-based assembly and map back all the data to it, what you can see is actually what's going on with these assemblies. So here you've got 3 microchromosomes from this PacBio assembly, from this nanopore-based assembly that was published recently. And you've got some tracks, you've got the repeat density, GC content, and then you've got the coverage of the nanopore reads, some Illumina reads, and some HiFi reads. And what you can immediately see, where I've highlighted with these red arrows, is along this chromosome, there's certain regions of the chromosome, like half this chromosome, nearly half this chromosome, the coverage of the PacBio HiFi reads just completely drops out, it disappears. But when you look at the nanopore read coverage, it stays relatively smooth along this region, doesn't drop out as much. And so this other chromosome over here, chromosome 30, the same thing, there's these regions where the PacBio HiFi data is really dropping to very low coverage, whereas it's much smoother in the nanopore with the nanopore reads. And what's striking is where this coverage is dropping out, it's not where the repeats are. So the repeat track, the ends of the chromosome on chromosome 16, you've got 80% repeats, but where this coverage dropout is occurring, there's like 10% repeats. It's very repeat poor. But what it does overlap with, if you look at the RNA-seq alignments, which is this second from bottom track, the region with the coverage dropout exactly overlaps where all the genes are in this chromosome. So we've got this problem where, for whatever reason, the sequence composition of this chromosome is preventing PacBio HiFi from working very well at sequencing it. And consequently, we get this dropout in read coverage. And I think that's really what's leading to the fragmentation of microchromosomes in these bird assemblies, is that the PacBio HiFi just doesn't work well in these gene-dense regions of the micros, which is obviously quite a big problem because the gene-dense regions are probably the regions we're most interested in. So we need to be quite cautious with these HiFi assemblies. And I think what it shows is that actually, if you just add in a bit of standard nanopore sequencing to your assembly, you can actually make a dramatic improvement to the microchromosomes just by having better representation in your assembly, because they don't suffer from this coverage dropout. And so what I've got another bird assembly which was done with nanopore, which if you want, we can have a quick look at now. So if you go back to the Git pod, and if we load up, so we go into the GUI and we click U to load up in PreText another map. And then we go up to the top here, click load map, and go back into maps and save states. And then we want to load up B-acropal2, which is this labeled as an ONT assembly. And we can load up the draft assembly of this one here. Okay, so this was one that we were curated at Sanger recently. It's very contiguous, but what's striking, so there's a lot less shrapnel, but we could actually find, so I've just adjusted the contrast here so you can see it better. So there's various chromosomes here, but if you look here, what's happened in this assembly is there's one scaffold up here. I'm just trying to find it for you. Move along. Here. So this scaffold looks quite a bit different to all the others. So here we've got, this is like a fairly normal looking scaffold. There's an assembly error here where the first chunk wants to go to the end. But then if we look at this scaffold, there's lots of individual blocks which have been joined to scaffolded together. And I think in this assembly, the scaffold has basically got a bit confused by the elevated background signal, and it seems to have joined small chromosomes together. So here, if we zoom in, we put the gaps on. Here you can see there's these drop-off points in HiC contacts actually correspond to gaps in the assembly as well. So, for example, here, this looks like one individual microchromosome on its own. If you look along the assembly, it's not hitting any of the other large chromosomes. So I'm looking off the diagonal here. It doesn't belong with any other chromosome. I go the other direction, check the other direction. And it's not hitting much of the shrapnel, and it's not hitting any other chromosome. So this is really classically looking like a single small chromosome on its own. very dense HiC contacts on the diagonal, and it doesn't belong with anything else. If you zoom in, you can actually see here, this is another useful signal. So at the end of the chromosome, at this end, you see this blob of contacts, which corresponds to the end of another adjacent chromosome here. And if you look along the assembly, you see other dots of black here. So this is actually likely a telomere sequence at the end of the sequence, which is being picked up by the HiC contacts. So we can see this is the one end of the chromosome. There's another one here, and as you see, this sharp drop off in contact. So this one's breaking here. And here.
Tom,
There's a half. Yeah.
can you just clarify if this assembly that we're looking at, this is our HiFi assembly, is it? Or the ONT assembly at the moment?
Yeah, so this is a nanopore assembly.
Yeah, so somebody was just asking if it's ONT only.
So I think this one was ONT only. The chicken one that was published was a mixture of HiFi and ONT, but this one was a de novo ONT as only assembly. I think this one came in through ERGA originally, from the ERGA pilot project, and there were some of the labs there were doing nanopore only assemblies. So that, yeah, it's a nanopore assembly only, and so that breaks there.
Do you know the assembler, Tom?
I think they did them with Next de Novo. So we've experimented with Next de Novo a bit, and that works quite well. And these, I'm pretty sure I checked this at the time because I curated this one at the time, and it was a Next de Novo assembly. I could double check that, but I think that's what it was. Again, here there's another possible break or flip here. So the main point though is that when we eventually break all these up, what I'll do now is I'm just going to show you the curated version of this assembly. If you load map, and then if you load the BGA24 primary curated.pretext, load that up. And then this is the curated version of the draft assembly I just showed you. And if we zoom in, we can see in total we've been able to find, where are we? So we've got 39 chromosomes in here. And this, I remember checking at the time, this corresponds to the expected karyotype for this species. And they're all quite large. So the smallest chromosome is nearly a megabase. Actually, it is 1 megabase long, I think. So they're all much bigger than what we see in these HiFi assemblies. One thing I will try, and I think is worth trying, is probably making, we haven't had time to do it yet, but making a reduced map of the smallest scaffolds below 20 megabases again for this type of nanopore assembly, and then having a closer look to see if we can see any other assembly errors in these micros. But the main point is that we get the karyotype relatively easily compared to curating the HiFi assembly. The chromosomes are much bigger and probably a lot more complete. We still have some repeat, which we can't place, which is at the end of this assembly. And this is just kind of hitting multiple chromosomes across the assembly. You can see the off-diagonal signal going to multiple locations. So we're still, it's not telomere to telomere, it's not gapless, but it is a significant improvement over what we see with the HiFi assemblies. So I would recommend, if you're doing bird assemblies, if you have the opportunity to go back to the lab and generate some nanopore data, I think it's likely going to be beneficial for your assemblies in the long run. Okay, what we can do is, I don't know if anybody has any more questions.
Even if it's not ultra long, right, Tom? So
yeah, I think it doesn't have to be ultra long. I think because of what we can see with the mapping of the HiFi reads and the nanopore reads back to the assembly, the nanopore assembly, because of I think the problem is mostly caused by the sequencing dropout of the HiFi reads in certain regions of the chromosome. So just having sequence coverage of that region with your nanopore reads, I think will enable you to actually assemble the chromosomes much better and make it much easier to curate. And you should have less missing content. At Sanger, we're currently sequencing 20 birds, I think, with nanopore that we've already generated HiFi data for. So we're going to run some sort of systematic evaluation of these HiFi versus nanopore assemblies for birds. We're slightly ahead of time. If there is any questions about any of the tools or things that we've looked at, either shout out or pop it in the Discord. If not, we can just do a quick wrap up and finish a bit early.
Comment on the quality of the HiC and how much HiC data you think, to be able to see to the level of detail that the map that we looked at together Because
Yeah,
that's
so that one was, I think it was slightly higher coverage, at least 50x, I would say, of HiC for that one. And we do see quite a bit of library to library variation as well in what it looks like in the maps. So there, I've also included, if we load up another assembly, there's one called, if we go into the, which one am I in? If you go in the, I'm not in the right window, sorry. If you load up Valval1 here, this one I think is, I've got a reduced map, which is the HAP1 first pass max 20 megs dot PreText. This map, which you can have a look at now, you still get the extra detail and you can spot errors in the micros using this approach, but it's not as dense as the other one. So it's slightly less clear. And so there is quite a bit of variation from library to library, and I think having more coverage for this, when you want to zoom in on these very tiny chromosomes, making these higher resolution maps, certainly having more HiC coverage will be beneficial. So I would aim for at least 50x. I think, I can't remember what the Genome ARC ones aim for. It might be more than 50x. I don't know if you know, Joe or Dom, but I think they typically do slightly higher coverage than the Sanger birds. But certainly I would say minimum of 50x, and more the better for this method. Again, because we've only just started playing about with it, we haven't done anything systematic yet to say what you would exactly need to be able to do it, but certainly the more the better, I would say. Yeah, so if there's any other questions, then fire away. If not, I would just do a quick wrap up. I can also actually show you there's another bird, we had another bird through. So this was a recently published assembly of the bustard, which was done. This is a next de novo ONT assembly. They got the full karyotype of the bustard, really nice high quality assembly, very, very complete, very few gaps in the assembly. There are a few assembly errors in it. So if you zoom in, we remade the PreText map of this nanopore assembly. You can see some errors here, which are the red arrows. So, for example, this sequence in the Z chromosome should be flipped. And this one on chromosome 13, the off-diagonal contacts here, this needs a flip. These are just errors in the macrochromosomes. So curation is still important for these nanopore assemblies. They're not kind of finished out of the box without any additional curation. But again, this got really nice assembly of the micros. The smallest chromosomes ranged from 2.9 to 680 kb. And if we compare this, so this was published, but there was also the same bird species came through Sanger. And I think, Joe, you did the curation for this one. So this is Joe's phase diploid assembly of the same bird. And these are, again, the smallest 11 chromosomes in Joe's version. And in this case, we got the largest one is around 2 megabases, but the smallest one is only 300 kb. So the micros, again, even after a substantial amount of curation effort, they're still substantially smaller than you get with the nanopore assembly. And I had a look at mapping the read data back to this nanopore assembly. So this is our hi-fi reads map back, and then at the bottom is the ONT data map back. And you can see, again, these big drop-offs in coverage in the hi-fi data. So at the beginning of like chromosome 16, for example, there's a lot of repeats at the beginning, which causes, for both technologies, the read mapping is elevated just from multiple mapping to various repeats. And then as you go along the chromosome, you see this big drop-off towards the end of the chromosome with the hi-fi data. It's dropping from like 30 to 40x to 0, close to 0. Chromosome 29, again, very low. And I don't think I included one, but the average depth across the macros is this red line, which is 56x depth across the large chromosomes. And all these micros have big chunks of them which drop substantially towards 0 in certain regions of these chromosomes. Whereas with the nanopore data, the drop is much less. And what's interesting again is you see that these, so here I've indicated chromosome 29, for example, which is very low coverage in the hi-fi data. It's not especially repetitive. If you look at the HiC data, it looks very smooth on the diagonal, and it's not hitting loads of other places in the assembly. So it's not, the coverage isn't dropping off because of repeats. Again, it's this problem with euchromatic regions of these micros with lots of genes where it's just not not sequencing properly with the PacBio. So there's another example of a nanopore assembly which looks quite good. Okay, so I've shown you a bit of data showing really that HiFi assemblies of bird micros are quite challenging, as probably most of you know who've tackled them before. We developed a tool called Microfinder, which enables us to fish out these small contigs which are associated or candidate microchromosome contigs from the shrapnel of an assembly, move them to the front of the assembly, and speed up the curation process. That's available on GitHub. We're currently working on the documentation and making that better, but you can have a go with that. The links are on the page in Discord, where I've added the information sheet. We're also developing this 2-pass curation method for birds, where we do an initial curation as best we can, split out the very small scaffolds, remake the HiC map, and then do a detailed curation of those small chromosomes to improve the accuracy. It also allows us to place more assembly content onto those chromosomes as well. Initial testing of this is looking quite good. There's an issue with PacBio HiFi in the gene-rich regions of the micros, and we would definitely recommend generating some nanopore data if you have the capability to do that as well as the HiFi data, which would probably help. Thank you for your attention. So if it got a bit confusing when I was trying to show you around the map, there's a lot to do on those maps, but hopefully it was useful. As David says, you can fill in the feedback and we can try and improve things for next time. And yeah, thank you for your attention. Get in touch if you need some help.
Final chance for questions, everyone. It's typing in the Discord, I can see.
Okay, yeah, we can hang on for a minute. That's fine. Sam thinks he's in the Matrix. Yeah, it does feel like that sometimes, Sam. You can stare at these things a lot, I think. Right. Okay, I think we can probably wrap up if everybody's happy.
Yeah, oh, there's more typing going on. No? Cool. Okay. Right. In that case, thank you all for coming. Thank you guys for running the session. And I hope to see you all in the next one. Goodbye.
Thanks a lot. Bye.
Cheers.